Welcome to Essential Mechanisms of Biochemistry. In this video, we are going to treat the pentose phosphate pathway, not as a list of reactions to memorize, but as connected to a system for capturing carbon, reducing carbon, rearranging carbon skeletons, generating reducing power, and producing 5 carbon sugars in the process. We begin with the reductive pentose phosphate pathway, commonly called the Calvin cycle. Reductive is the chemically informative name. Carbon dioxide is captured and then reduced to carbohydrate using ATP and NADPH, generated by the light reactions. Before following the reactions, however, we need a vocabulary of carbon skeletons. On the left are aldoses, whose open-chain carbonyl is an aldehyde. On the right are ketoses, whose carbonyl is a ketone. The smallest metabolically central aldose is glyceraldehyde 3-phosphate. Its ketose partner is dihydroxyacetone phosphate. 4, 5, 6, and 7 carbon sugars are created by repeatedly joining, splitting, and transferring fragments derived from these smaller units. Carbon fixation begins with ribulose 1,5-bisphosphate carboxylase oxygenase, Ribisco. This enzyme first removes a proton to form an ene-diolate from ribulose 1,5-bisphosphate. That ene-diolate is the carbon nucleophile. It attacks carbon dioxide and produces a 6-carbon carboxylated intermediate. Water then adds to the carbonyl. Hydration destabilizes the intermediate and prepares the carbon-carbon bond between C2 and C3 to break. The cleavage produces 2 molecules of 3-phosphoglycerate. The reaction is thermodynamically favorable under standard biochemical conditions. The difficulty of carbon capture is therefore not that carbon dioxide fixation is intrinsically uphill. The difficulty is kinetic. Carbon dioxide is dilute, relatively unreactive, and must be distinguished from oxygen. The newly produced 3-phosphoglycerate contains a carboxylate, and carboxylates are poor electrophiles. Before that carbon can be reduced, the cell must activate it. The carboxylate oxygen attacks the gamma phosphate of ATP as ADP leaves. This creates 1,3-bisphosphoglycerate, an acyl phosphate. This is most definitely an ATP investment, not ATP production. ATP raises the free energy and electrophilic reactivity of the substrate so that reduction becomes chemically accessible in the next step. And now the activated acyl phosphate is reduced by NADPH. A hydride is transferred from the nicotinamide ring of NADPH to the carbonyl carbon of 1,3-bisphosphoglycerate. The tetrahedral intermediate collapses, phosphate leaves, and glyceraldehyde 3-phosphate is formed. This is the decisive step, reductive step, in the Calvin cycle. Carbon dioxide entered as highly oxidized carbon, but after ATP activation and hydride transfer, we now have an aldehyde, a genuine 3-carbon carbohydrate building block. We now leave the reduction phase and enter the rearrangement phase of the pentose phosphate pathway. The pathway already possesses a 3-carbon carbohydrate. The remaining problem is architectural. How can 3 carbon units be redistributed into 4, 5, 6, and 7 carbon sugars until the original 5-carbon carbon dioxide acceptor can be regenerated? It is important to note that the reductive phase of the pentose phosphate pathway, i.e. the Calvin cycle, is unique to photosynthetic organisms. However, the rearrangement phase and the oxidative phase, as we will subsequently see, is not unique to just photosynthetic organisms. Now, glyceraldehyde 3-phosphate is an aldose because its carbonyl is on C1. Dehydroxyacetone phosphate is a ketose because its carbonyl is on C2. An enzyme base removes a proton adjacent to the carbonyl, producing an enediol or enediolate intermediate. Reprotonation at the opposite carbon relocates the carbonyl. No carbon atoms are added or removed. The oxidation state is unchanged. The phosphate remains in place. Only the position of the carbonyl changes. But now we bring in aldolase, in which a 3-carbon sugar plus a 3-carbon sugar gives us a 6-carbon sugar. A lysine side chain attacks the carbonyl of dehydroxyacetone phosphate, producing a carbonyl amine. Proton transfers convert the hydroxyl into water, which leaves to produce a Schiff base, an iminium ion covalently linking the substrate to the enzyme. Deprotonation produces an enamine. This reverses the usual polarity of the carbonyl partner. The alpha carbon becomes nucleophilic and attacks the carbonyl of glyceraldehyde 3-phosphate. After carbon-carbon bond formation, water attacks the iminium ion, the carbonyl amine reforms, and the enzyme lysine is released. The product is fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate now contains phosphates at both ends. Hydrolysis removes the phosphate at C1 to form fructose 6-phosphate and inorganic phosphate. This is not the reverse mechanism of a kinase. A phosphatase uses water to hydrolyze an existing phosphate ester. A kinase uses ATP as an activated phosphoryl donor to construct one. Phosphorylation provides charge, solubility, recognition, and metabolic confinement. Here, selective dephosphorylation changes which enzymes can recognize the sugar and permit the next carbon transfer steps. Here we introduce transketolase, which initiates a 2-carbon transfer. Transketolase uses thiamine diphosphate, derived from vitamin B1, to transfer a 2-carbon fragment. The aminopyrimidine and thiazolium portions of THDP cooperate to generate the reactive ylide. The positively charged thiazolium ring stabilizes carbanion character adjacent to it. The THDP ylide attacks the carbonyl of the donor ketose, fructose 6-phosphate. After bond cleavage, a 2-carbon fragment remains covalently attached to THDP while erythritose 4-phosphate leaves. The 2-carbon fragment then attacks glyceraldehyde 3-phosphate. Product release yields xylulose 5-phosphate and regenerates the cofactor. The central rule is simple. Transketolase moves an activated 2-carbon glycolaldehyde equivalent without changing the overall oxidation state. Furthermore, we introduce transaldolase, associated with 3-carbon transfers. Don't mix this up with aldolase. Aldolase combined 3-carbon sugar and a 3-carbon sugar to form a 6-carbon sugar, whereas this is a 3-carbon transfer. A lysine attacks fructose 6-phosphate and forms a Schiff base. Cleavage releases glyceraldehyde 3-phosphate while leaving a 3-carbon dihydroxyacetone fragment covalently attached to the enzyme as an enamine. That 3-carbon nucleophile attacks erythrose 4-phosphate. Hydrolysis then releases sedu-heptulose 7-phosphate and regenerates the lysine. Compare the two carbon transfer enzymes. Transketolase transfers 2 carbons using THDP, and transaldolase transfers 3 carbons using a lysine shift base. And now we return to a different version of aldolase, only found in photosynthetic organisms in this case, in which 3 carbon plus 4 carbon sugars will give us a 7 carbon sugar. Dihydroxyacetone phosphate again forms a shift base with an active site lysine and becomes an enamine nucleophile. This time the acceptor is erythrose 4-phosphate rather than glyceraldehyde 3-phosphate. Carbon-carbon bond formation therefore creates a 7-carbon product, cetoheptulose 1,7-bisphosphate. The general rule has been expanded. A 3-carbon ketose can be joined to either a 3-carbon or 4-carbon aldose, in this case, using the same covalent catalytic strategy. Furthermore, cetoheptulose 1,7-bisphosphate is hydrolyzed to cetoheptulose 7-phosphate, an inorganic phosphate, using cetoheptulose 1,7-bisphosphatase. As with fructose 1,6-bisphosphatase, this is a strongly directional hydrolysis under cellular conditions. The enzyme is not running a kinase backward. It is using water to remove a phosphate. This dephosphorylation commits the 7-carbon product to the regeneration network and prepares it for the next 2-carbon transfer. And now we have the second transketolase reaction mechanism. Here again, the THDP mechanism is unchanged. The ylide attacks the donor ketose, cetoheptulose 7-phosphate. Cleavage releases ribose 5-phosphate and leaves a 2-carbon fragment attached to THDP. That fragment attacks glyceraldehyde 3-phosphate and produces xylulose 5-phosphate. The products are therefore 2 different 5-carbon sugars. The chemistry has redistributed 10 carbons into 2 pentoses without oxidation or reduction. And now enter phosphopentose epimerase. Xylulose 5-phosphate and ribulose 5-phosphate have the same carbon skeleton, the same carbonyl position, the same oxidation state, and the same phosphate position. They only differ at carbon 3. A divalent metal ion stabilizes the ene diolate produced when a proton is removed from C3. Repronation from the opposite face inverts that single stereocenter. This is epimerization, selective inversion at one stereocenter while the rest of the molecule is conserved. Now enter phosphopentose isomerase. Ribose 5-phosphate is an aldose with the carbonyl at C1. Ribulose 5-phosphate is a ketose with the carbonyl on C2. An enzyme base removes the alpha proton. An ene-diol or ene-diolate forms, and repronation relocates the carbonyl. Once again, no carbon is gained or lost, and no oxidation state changes. The pathway is editing functional group position so that multiple pentoses converge on ribulose 5-phosphate. And with phosphoribulokinase, we regenerate ribulose bisphosphate. The final step here regenerates the carbon dioxide acceptor. Ribulose 5-phosphate contains 1 phosphate. Ribisco requires ribulose 1,5-bisphosphate. So phosphoribulokinase transfers the gamma phosphate of ATP to the C1 hydroxyl. An enzyme base activates the alcohol, The oxygen attacks phosphorus in line, and ADP leaves. This is ATP-dependent phosphoryl transfer. Contrast this with the two bisphosphatases. Uh, Hydrolysis removes a phosphate because phosphate ester cleavage is most definitely favorable, whereas installing a new phosphate requires an activated donor. The cycle has now returned to ribulose 1,5-bisphosphate and is ready to capture another carbon dioxide. Additionally, another important route needs to be highlighted with phosphoglucose isomerase. Fructose 6-phosphate, our transfer sugar, is a ketose with its carbonyl on C2. Glucose 6-phosphate, our stored sugar, is an aldose with its carbonyl on C1. The mechanism is the same enediol-mediated carbonyl migration seen in triose phosphate isomerase and phosphopentose isomerase. The carbon skeleton, oxidation state, phosphate position, and pre-existing stereochemistry are conserved. Only the carbonyl position changes. Because no ATP is consumed and no strongly exergonic hydrolysis occurs, this reaction is readily reversible and its direction responds to metabolite concentrations. However, the linear structures that we have used in the mechanisms provided are only one part of the molecular population of sugars in water. Fructose 6-phosphate easily interconverts between open chain and fructofuranose forms. For ketoses, the anomeric carbon is C2. Phosphorylation at C6 blocks the C6 hydroxyl that unphosphorylated fructose can use to form a pyranose ring, leaving the C5 hydroxyl to form the furanose. Glucose 6-phosphate exists predominantly as alpha or beta glucopyranose, which interconvert through the very small open chain population. Ring opening restores the C1 aldehyde. Ring closure creates the anomeric stereocenter at C1. The major conceptual point here is dynamic equilibrium. Even a tiny open chain population can connect and replenish the much larger cyclic populations. Finally, we end with turning the pentose phosphate pathway around conceptually. The reductive branch used ATP and NADPH to build carbohydrate from carbon dioxide. The oxidative branch begins with glucose 6-phosphate and oxidizes it to generate NADPH and a 5-carbon sugar. The same carbohydrate network can therefore either consume reducing power or produce it, depending upon the direction and combination of reactions used by the cell. Glucose 6-phosphate dehydrogenase begins the oxidative branch. The cyclic hemiacetal opens in the enzyme active site, exposing the C1 aldehyde equivalent. A hydride is transferred from C1 to NADP+, forming NADPH. Oxidation converts the hemiacetal carbon into the carbonyl of 6-phosphoglucano-delta-lactone. The 6-carbon skeleton and phosphate are conserved. The oxidation state, however, changes, and reducing power has been harvested. This is the first committed step of the oxidative branch and the first of two NADPH producing reactions. With 6-phosphoglucanolactonase, we begin with the product of the former reaction, a lactone, which is an intramolecular ester. Water attacks the ester carbonyl, producing a tetrahedral intermediate. Collapse of that intermediate breaks the acyl-oxygen bond and opens the ring, yielding 6 phosphogluconate. No redox chemistry occurs here. The carbon skeleton and oxidation states are conserved. The enzyme simply accelerates ester hydrolysis and converts a reactive cyclic ester into the stable open-chain carboxylate required for the next oxidation step. And now the final oxidation reaction combines oxidation and decarboxylation. A hydride is transferred from 6-phosphogluconate to NADP+, producing a second NADPH and a beta-keto acid intermediate. Beta-keto acids decarboxylate readily. Carbon dioxide leaves, generating an ene-diolate that is protonated and tautomerizes to ribulose 5-phosphate. The carbon count falls from 6 to 5. One carbon is sacrificed as carbon dioxide, but 2 NADPHs have now been produced in total, and the product, ribulose 5-phosphate, can enter the same rearrangement network we followed earlier. That closes the conceptual loop. The reductive pathway consumes NADPH to build carbohydrate, while the oxidative pathway oxidizes carbohydrate to generate NADPH and pentose phosphates. The pentose phosphate pathway is not one rigid sequence. It is a reversible carbon rearrangement network connected to two directional redox molecules. In photosynthetic carbon fixation, ATP and NADPH drive carbon dioxide toward carbohydrate. In the oxidative branch, glucose 6-phosphate is oxidized to produce NADPH and pentose phosphates. Between them lies a shared chemical language. Ene-diols move carbonyls, shift bases, join and transfer carbon. THDP carries 2 carbon fragments. Phosphatases remove phosphate, kinases install it, and dehydrogenases exchange hydride with nicotinamide cofactors. Till next time, Crip.